As a result of its developmental activities spanning over many years and supported by economic development fundings, Histopathology Ltd has designed the DominoStainer, an automated immunostaining instrument. George Sekeres, managing director of the company, and his French colleague Michel Delage prepared the patent description of the solution on which the instrument is based. A French company, Inodiag, and Histopathology Ltd acquired the patent exploitation rights for two different applications. This latter, based on market demand, carried on development and created several functioning prototypes, which made this modern technology useful both for processing tissue microarray samples and for small routine laboratories. The basic principle of the instrument consists in sealed incubation chambers, arranged next to each other in a domino-like fashion, combined with connecting valves and a pumping system, controlled by built-in touchscreen electronics. Prior to applying the reagents and starting the immunohistochemical protocol, it is recommended to prepare the pipes and chambers of the instrument for the procedure. Start DominoStainer using the switch on its rear panel. The main menu buttons, located under the touchscreen, can be used to switch between the setup and the control windows. When all 5 chambers are used, the optimal reagent volumes calculated for one staining cycle are the following. In case of Protocol 1, which is a manual primary antibody application, fill reagent vial R1 with 3 ml of distilled water. However, in Protocol 2, the automatic antibody application, with 3 ml of primary antibody, filling the additional reagent vials is the same for both Protocol 1 and 2. 3 ml of secondary antibody in reagent vial R2. 3 ml of chromogen substrate system in reagent vial R3. Approximately 10 ml of hematoxylin solution in reagent vial R4. Approximately 10 ml of tap water in reagent vial R5. A minimum of 100 ml of buffer in reagent container R6. A minimum of 100 ml of distilled water in reagent container R7. Each chamber must contain a blank slide both in suction and wash phases, and also in standby mode. Press F5 on the main menu bar. Select the System Stop mode on the touchscreen. Then press F2 on the main menu bar. Depending on the required protocol, press the buttons assigned to the specific reagent vials under suction on the touchscreen, one by one, to actuate suction of the reagents. Proceed one at a time, waiting for each particular cycle to be completed. Go back to F5 on the main menu bar. Press System Start Application on the touchscreen. Press F2 on the main menu bar. Then perform the rinse step for the individual chambers under the rinse item, proceeding one at a time, waiting for each particular cycle to be completed. Staying in F2 menu, perform the rinsing of the intermediate network by pressing the DR Start button on the touchscreen. Select F5 menu on the main menu bar, which contains settings of the individual incubation chambers. Depending on protocol selection, press P1 or P2 buttons assigned to the appropriate chamber on the touchscreen. Open the first chamber. Remove the blank slide. Wipe the inside of the chamber dry. If Protocol 1 was selected, add 150 microliters of optimally diluted primary antibody into the chamber. In case of Protocol 2, manual application of primary antibody is unnecessary. It will be introduced by suction from reagent vial R1 at the protocol start. Wipe the slide to be stained around the tissue section. Carefully load it in the chamber with its tissue-containing side down. Then close the chamber. Perform this action one by one for all the 5 chambers, or if there are fewer sections, for the chambers to be used. After closing the chambers, press the Start-Stop button on the touchscreen under their corresponding label. The progress of the selected protocol can be monitored under this F5 menu, as well as via a process indicator table by switching over to F1 menu. When the staining run is complete, press the Stop OK buttons, one by one, for each chamber in the F5 menu on the touchscreen. Now the chambers containing the sections can be reopened and the stained sections can be removed. In case you wish to perform another immunohistochemical staining, the system must be rinsed through with buffer. Maintenance cleaning of the DominoStainer must not be forgotten either. Insert blank slides in the chambers and close them. Distilled water must be used for cleaning the system. Perform the necessary post-treatment of the stained sections, that is, dehydrate and cover them. After that, the samples can be directly examined. During the evaluation, conclusions as to pathological and healthy conditions can be derived by analyzing the light microscopic image. On the basis of the biological parameters of pathological, for example, malignant tumor cells, not only the diagnosis, but also the outcome of the pathological process can be predicted.
Histopathology. In the service of diagnostics.